All right, so we're going to get started. Our next speaker is Alex Young, who we're really excited to have here. Alex did his PhD in genomic medicine or statistical genetics. What's the actual?
Genomic medicine and statistics, but well, the title doesn't really matter that much.
I was close. And then he's been doing a postdoc with Augie Kong, first at Genetics and now at the Big Data Institute at Oxford. They worked together on a paper on genetic nurture that Augie led, and Alex played an important role on, which was when it was first being presented was very controversial and people weren't quite sure it was being interpreted right. By now, everyone has been converted and is widely recognized as one of the major contributions in statistical genetics within the last couple of years. Alex has been working on family methods since then, including developing a new method of estimating heritability that he's going to be talking about. And this area that Alex is a leader in, the family-based methods, is clearly one of the most important areas at the current cutting edge of statistical genetics. So we're thrilled to have Alex here.
Thanks. Yeah, there is some irony that, all right, thanks. There is some irony that the family methods are now the cutting edge of statistical genetics, given that that's what everyone used to do before GWAS. So it's really, in some sense, going back to the origins of sort of genetic analysis. So this makes sense, really. Genetics is about inheritance and families, and so that's part of what I'll be talking about. And all of the family-based methods are really important for disentangling genes and environment. So when you have correlations between genes and environment, the most effective way to disentangle them is using family data because of the randomization of genetic material that occurs in the family, and that's sort of going to be a theme of the lecture. So why should we care about gene-environment correlation? There are various reasons, but I think the most sort of pressing reason that the field is concerned about is that it biases GWAS effect estimates. So if you consider a phenotype, I'm just going to, for the theoretical derivations, I'm just going to consider a toy phenotype that's determined by the direct causal effect of a genetic variant when you inherit it, and then some environmental influence. Then if that genetic variant G is correlated with the environmental influence on the trait E, your estimate of the causal effect of the genetic variant is going to be biased in proportion to that correlation. And there are two main sources, I would say, of gene-environment correlation, although they're perhaps not completely distinct. They're perhaps the same thing at different scales. But the type of gene-environment correlation that people have worried about traditionally in genome-wide association studies is sort of broad-scale population stratification. So this is a map of the genetic clusters of the people of the British Isles, though it's actually probably a map of what the genetic clusters were in the past, because it's not necessarily reflecting current genetic variation in Britain. But you can see that the genetic clusters correlate with geography, and many kinds of environmental effects also correlate with geography. I mean, if you're just going north to south, then clearly sun exposure or something like that is going to correlate with geography. So if you have a genetic variant that is differentiated with latitude, it's a high frequency in the south versus the north of Britain, then that genetic variant is going to become correlated with any trait that is affected by, say, sun exposure. And people have developed methods to deal with this kind of stratification that work pretty well for the kinds of analysis typically done in GWAS, principal components and linear mixed models, which I think people talked about principal components, at least in the course. And this kind of stratification can be adjusted for much more easily than the second kind of stratification I'm going to talk about, which is genetic nurture. So typically when you think of the effect of a genetic variant, you think you inherit some allele from your parents. then that allele exerts a biochemical effect on you throughout your development, and then that affects your trait. So that's the standard sort of genetic effect that people think of. But any allele that you inherit was also present in at least one of your parents. So if you imagine this allele tends to increase educational attainment, you inherit it, so it can have some direct effect on you, increasing your educational attainment. But if it was also present in your parents, it could have increased their educational attainment. And by increasing their educational attainment, that changes the environment in which you develop. And it can thereby influence your educational attainment not only through inheritance, but through the environment provided for you by your parents. In this kind of gene-environment correlation, I guess it's called like passive gene-environment correlation in the behavior genetics literature, it's very difficult to adjust for based on the kinds of data that are present in genome-wide association study data typically. And that's because if you simply correlate in individuals' alleles with their traits, then that correlation can be driven not only by the direct effect of inheriting it, but also by the indirect effect that comes through the family environment. And these kinds of effects are sort of indistinguishable on the basis of just correlations between alleles and traits. So there's a question of like, to what extent are the associations, the effects that we estimate in GWAS, to what extent are they actually picking up effects that are coming through the family environment rather than simply the direct causal effect of inheriting that genetic variant? It's not only parents that can exert this indirect genetic effect, it can also come from other relatives. And any sort of relative that has an indirect genetic effect is going to contribute to this gene-environment correlation thing because relatives' genotypes are correlated with your own. So this correlation will thereby contribute towards the GWAS effect estimate. So beyond parents, siblings are a natural relative that might influence your trait. And if there's a genetic component to that, then some of the GWAS effect estimate can actually pick up like the indirect genetic effect coming from the sibling, the influence of your sibling on your phenotype. And beyond parents, grandparents can clearly have a direct effect, but actually kind of a more interesting pathway is that your grandparents influence your parents and your parents influence you. So in some sense, these indirect effects can accumulate over like generations. And in some sense, if you go back enough generations, then it can be more like a classical population stratification effect than a nuclear family type indirect effect. And in some sense, population stratification, indirect effects from close relatives is like a very fine-grained form of population stratification. So they can kind of be the same phenomenon, but at different scales. Although obviously you get environmental stratification at geographic scales, it's unrelated to genes, but some of what we traditionally think of as like population stratification could actually be related to these indirect genetic effects iterated over many generations. So these phenomena are related and not completely distinct, but just happening at different scales and through different mechanisms. So when genes and environment are correlated, the sort of standard way of decomposing trait variation or modeling a quantitative trait is sort of missing some of the story, in my opinion. So the standard additive model, you'd have a phenotype determined by some direct effect of a genetic variant and then an environmental variable, and then you assume that they're uncorrelated, or you kind of define the genetic effect to be the regression of the phenotype onto the genotype, and then the environment is everything that's orthogonal to that. But as I was saying before, when you have these indirect genetic effects and population stratification, that regression can be picking up some of those G by E correlations. It's not simply picking up the direct causal effect. So that assuming that they're uncorrelated or defining the genetic effect in that sort of regression way rather than like a causal way, then you decompose the phenotypic variance into just two components, a genetic variance component and then whatever is like orthogonal to that or assumed to be orthogonal to that. But I would argue that this kind of misses, especially when you're thinking about social science type traits, this kind of is an incomplete picture of the relationship between genes and environment that can sort of lead you astray. And the way to think about it, I think, is to take advantage of the randomization of genetic material in the family to understand the relationship between genes and environment and have a more general kind of picture of the different components of phenotypic variation. So this is a directed acyclic graph showing like the determination of offspring genotype. So you're sort of showing the sort of causal influences. Then the offspring genotype is determined by the parental genotype, And then the random segregations that happen in the parents during meiosis, the production of sperm and eggs. So you inherit randomly the red or the blue allele from your mother, and the red or the blue allele from your father. And so what that means is conditional on the genotypes of the parents, all that's left to determine the genotype of the offspring is essentially like these random coin tosses that happen in meiosis. And that means that any dependence between offspring genotype and environment must actually flow through the parental genotype. So you can break this dependence between offspring genotype or the phenotyped individual's genotype and their environment through conditioning on the parental genotype. And through the conditional independence properties that derive from the randomization of genetic material during meiosis, one can split the environmental component of the trait into a component that is correlated with parental genotype. And this eta coefficient gives the sort of strength of this correlation between the environmental influence and the parental genotype. And the parental genotype here is defined as the sum of the maternal and paternal genotypes. And then a residual environmental component that's uncorrelated with both the parental genotype and the offspring genotype. And then that allows you to write the phenotype in a way where you have an environmental component, this residual environmental component that's uncorrelated with the phenotyped individual's genotype and their parent's genotype. And this is sort of a more general way of writing a trait that takes into account gene-environment correlation in this term. And this term will capture the indirect genetic effects from the parents and some fraction of the indirect genetic effects from the other relatives. And this then leads to an easier way to sort of understand how to estimate direct genetic effects in an unbiased way. So yeah, that's just written out. So the residual environmental components uncorrelated with genetics. So you sort of, it's a more general way of writing the trait than the standard G plus E. You have an E that's split into a bit that's correlated with genetics and a bit that's uncorrelated with genetics in some sense. And if you write the trait in this way, it's actually simple to see what the standard sort of GWAS estimate is giving you and then what a family-based association method is going to give you. So the standard GWAS estimate is not going to be estimating just the direct effects, it's going to be estimating the sum of the direct effect in this eta term, which is capturing the indirect effects from relatives and other forms of gene-environment correlation. However, if you jointly regress the phenotype onto the proband and parental genotype, that will give you an unbiased estimate of the direct causal effect of the genetic variant. It doesn't necessarily give you an unbiased estimate of the indirect effects from the parents because this eta term can be capturing other forms of stratification and also sort of mating, but it does give you an unbiased estimate of the direct genetic effect. The main problem with this is there's not a lot of data where you need the genotype information on the parents of all your phenotype people to do this. And that, yeah?
Sorry, I just want to understand, so the G parents is just the direct sum of the genotypes of the paternal and maternal?
Yeah.
So there's no consideration of dominance effects or...
So this is assuming an additive model.
Okay.
You can do things with non-additive models, but it becomes very complicated.
Oh,
I mean,
okay,
yeah.
Like we discussed, but okay, so there's it's direct combination.
Yeah. So this method hasn't been widely applied because of the lack of data with genotype parents. So a more common method that has been applied is using sibling genotypes. So this can also get you something close to an unbiased estimate of the direct causal effect of a genetic variant. And this is because genetic differences between siblings are due to this random segregation in the parents. If you take two siblings and you consider the father's genotype, you've got two copies of each allele, each variant. Then you randomly inherit one or the other, so they can share none, they can share one, or they can share both, and whether they do is just due to these random segregations in the parents. So the genetic differences between siblings are random and uncorrelated with environmental effects on the trait. And also, this difference between sibling genotypes is uncorrelated with the parental genotype. And that's easy to see by symmetry because two siblings, the siblings' genotypes must be symmetrically correlated with the parental genotype. So the difference between the sibling genotypes is therefore uncorrelated with the parental genotype. And if you put that in the context of the... this doesn't seem to be very reactive, I have to say. If you put that in the context of this, then you can see that it's not picking... if you're regressing on the difference in the sib genotypes, you're not picking up any of this eta term that captures gene-environment correlation because they're symmetrically related to the parental genotype, so the difference is uncorrelated with that. So if you regress the phenotype onto the difference in the siblings' genotypes, that will give you something close to an unbiased. In some cases, it will give you an unbiased estimate of the direct effect. Sometimes people regress the difference of the sib genotypes onto the difference in the genotypes, but it's not necessary. You can't just regress phenotype onto the genotype minus the sib difference to also get the same expected estimate. However, because you're subtracting the sib genotype, if the sib genotype is affecting you through the environment, an indirect genetic effect from the sibling, then there is going to be some bias that comes from that indirect effect from the sibling. So it doesn't give the sibling method doesn't give us unbiased estimates under its general conditions. It's a method that uses parental genotypes, but it does pretty well. It removes classical forms of population stratification and indirect genetic effects from parents, but it doesn't remove indirect genetic effects from siblings, and it can actually lead to a biased estimate. So if you do the sibling method to try and estimate the direct causal effect, and you do the parental method to estimate the direct causal effect, and there are differences between the two, that could be evidence for indirect genetic effects between siblings. Okay. So, yeah?
Just, that might be a silly question, but this all hinges on the randomness, right, of what you inherit. How do we know for sure that that's completely exogenous of environment? Is there?
Well, no, I guess we don't, but I mean, it's probably about as robust an assumption as you ever get in the biological sciences. Like, you do get some deviations from like random inheritance due to like things. Some things have like meiotic drive and that, but they're probably very rare. It's hard to detect deviations from like the Mendelian inheritance if you actually look carefully, but there can be issues with ascertainment because some genotypes won't be observed if they're like lethal, for example. I mean, there might be some environmental variables that affect like meiosis, but I don't think anyone's ever really found them as far as I'm aware. Yeah?
I'm just having trouble with the covariance between the difference in siblings. Because if the parents are homozygotes, then we know that the difference between the siblings is 0, right? At least consider one allele.
Sure, yeah. I mean, you have to have genetic variation in the parents at a site for this. If you don't have any genetic variation in the parents, then you don't have any power. But I mean, you're generally looking over like a sample where you have variation, and there will be some.
I'm just thinking that whether the parents have both the same allele or different, then you might have differences in siblings or not. So there's some information there.
Well, there's essentially no variation in the SIB difference. If both parents are homozygous and there's no variation in the SIB difference, you don't have any power. But you're generally looking in a sample of parents that will vary. So maybe some of them are homozygous like that, and then there's no information from them. But it won't be all the parents being homozygous unless you're like looking at a very rare variant, and you don't really have any power.
We can talk more later.
Okay. This is sort of the background to, there's a question of how important are these kind of indirect effects from relatives in human traits, and this was a paper that I worked on in Iceland with Augie, where we were looking at it specifically for educational attainment, trying to figure out the influence of parental indirect genetic effects on educational attainment. And no, there seems to be quite a lag. Okay, so we took the educational attainment polygenic score derived from the EA-GWAS paper in 2016, so it's a bit out of date now, like the current one explains a lot more variation. But then, if this is in Icelandic data, if you just do the standard regression of the educational attainment or the polygenic score, you see that the R-squared is about 5%. But if you use this method where you're using parental genotypes to estimate the direct causal effect of the education polygenic score, that R-squared drops to around half of that. So that's sort of showing that some of the predictive power of the polygenic score is not coming from the direct causal effect of those variants, but at least it implies that at least some fraction of that is coming through indirect genetic effects from parents. This deflation in the R-squared within a family can also be due to removal of associative mating from it. So we had to do some extra work to adjust for that. And then in doing that, we got an estimate that the indirect effect from the parents is around 1/3 of the size of the direct effect. So it's fairly substantial. And it's worth remembering that in terms of variance explained, that can be a bigger effect than you anticipate, because the sort of estimate of the standard GWAS estimate or the standard regression estimate of the effect of the polygenic score, it's like the direct effect plus the nurturing effect, and then the variance explained is proportional to the square of that. So the effect of the indirect effects in inflating the variance explained is something more like 1 + 1/3 squared, not just like the 1/3 doesn't sound like a lot, but once you square the effect size, it can actually contribute a lot to the variance explained by the polygenic score because of the correlation, the gene-environment correlation, essentially. So then we also applied the educational attainment score to look at the variance explained. The variance explained in age at first child by the educational attainment score, and that was around 1%. But again, we saw that a big fraction of that was likely to be due to the indirect effects from the parents. And this is important because the age at which you have your first child is a fertility trait that affects human evolution. And this fits in with another paper that I was involved in where we showed that there was a decline in the mean education polygenic score in Iceland over the 20th century. So this is showing that part of that effect on fertility of the education score is actually mediated through the family environment. So these indirect genetic effects from relatives are affecting human evolution, need to be incorporated into evolutionary models. We also found some effect of the education score on this sort of composite health trait, and that some of that was going through the indirect effects from the parents. So these are also relevant to health. And when you're looking at the effect of polygenic score on one trait, derived from one trait on another trait, that's sort of telling you something about these genetic correlations. So in some sense, this is saying that the genetic correlation between EA and age of first child, that's actually partially mediated through the family environment. It's not, so to speak, directly genetic. Yeah?
I was curious, in that paper, did you do an analysis where you looked at polygenic scores of, let's say, age at first child, and then decompose it this way? I know you didn't report that in the paper, but did you do that?
So I don't think we had the age at first child score when we were doing this paper, but we have done that analysis, yeah, and you see a similar, I mean, the age of first child score and the education score are pretty correlated, I think, for one. So you see somewhat similar results. So yeah, that's just a point that these genetic correlations can be driven by these indirect effects through the family, not simply the direct effects. Then there's a question of differences between the effects of mothers and the effects of fathers, and I think this is a good example of how this genetic information can actually be used to investigate what happens in the environment, the family environment, because you might question, are mothers or fathers more important for like the health or the education of their offspring? And you could potentially investigate that through genetic variables. And we didn't have a lot of power to do that, but we did see some evidence, not statistically significant, for greater contribution from mothers for educational attainment at age of first child. For this composite health trait, we did actually have statistical significance, so you can see that the genetic contribution of mothers to some sort of health variables of the offspring is through the environment is probably greater than that of fathers. Then there's the question of the indirect effects from siblings. So we use this design where you regress the phenotype onto the genotype of the phenotyped individual, their sibling, and the parents. And this is actually a very robust method to estimate the effect of indirect genetic effect of siblings. And we found just about statistically significant effect of the sibling education polygenic score on the other sibling's trait. So that's some indication that these sibling effects can be real and important for a trait like educational attainment. And one of the interesting things about indirect genetic effects from siblings is that they necessarily lead to a violation of the equal environments assumption of the MZ, DZ twin design. And the reason for this is if your environment is partially determined by the genotype of your sibling, and if your sibling is genetically identical to you, the environment for monozygotic twins is more similar because they're genetically identical than the environment for dizygotic twins because they're genetically distinct. So this analysis here implies some bias in twin estimates of educational attainment. And that brings me on to heritability and what all of this gene-environment correlation stuff means for heritability and some of the methods that have been developed to estimate heritability from genomic data and other things. And I think it's important to define heritability as a fraction of trait variation due to direct genetic effects. And this makes sense to me conceptually, but it's also in concordance with the way people have thought about it in behavior genetics in the twin literature. You can define whatever variance component you want, but just make sure it's the same one as the other method if you're going to compare them. People don't always seem to do that. And it makes sense conceptually because from the perspective of the individual whose trait is affected by the indirect effects, although they are real genetic effects, but if your trait is affected by your parent's phenotype and that's partially genetically mediated, that's still an environmental effect from the perspective of the individual whose trait is affected. Yeah?
I think it's just worth pointing out that this is consistent with the way that we've been talking about heritability and causal effects of genotypes. Is the hypothetical experiment of changing your own genotype, but you're not affecting your parents' genotypes when you do that experiment.
Yeah, very consistent with that, yeah. Okay, so this way of writing the trait that splits the environment into a component that's correlated with parental genotype and uncorrelated actually leads to a fairly elegant variance decomposition that is more general than standard quantitative genetic variance decomposition, although no one really seems to have paid that much attention to this, but I think it's actually a better way of decomposing the phenotypic variance than the standard way, partially because it's hard to estimate these parameters based on current data. But using the fact that this residual environment is uncorrelated with the phenotyped individual's genotype and their parents, you can then decompose the phenotypic variance into a genetic component that gives you the heritability when normalized by the phenotypic variance. That's the variation explained by the direct effects. Then there's this component, which is the variance of the environmental component that's correlated with the parental genotype. So that's capturing the variance explained by indirect genetic effects from parents, but it's also capturing other sources of gene-environment correlation, such as population stratification and potentially also assortative mating. And then a third component that's due to the covariance between genetic and environmental influences on a trait. And then finally, the variance of the residual environmental component. So that's the component that's uncorrelated with the parental and phenotyped individuals. Yeah.
Have you looked to explore interactions between the child's genotype and the parental genotype? And the second question is, why would it fall in this model? Would it capture by G or VG?
Yeah, so we did actually look for an interaction between the offspring and parental polygenic score and educational attainment, and we didn't find anything. I mean, I'm sure that there is. Nothing's ever really linear, right? But the power is probably pretty limited for that, especially with that polygenic score. Maybe the new one would have more power. But if you only have a noisy estimate of the underlying genetic component, and then you're looking for interaction between two noisy estimates, then your power is pretty limited. But I imagine there is some non-linearity in there somewhere, but it's not really present. Current levels of polygenic score prediction. Well, maybe it is based on the new one. We haven't looked at that. Where that would fall, I haven't really thought about that, but I have a feeling it might be sort of split between the two different effects. I'd have to think about that a bit more. But based on the stuff that we've been doing, I don't think it's particularly important, but if we got a full genetic component, then we might want to worry about it more. So this gives you a decomposition of the phenotypic variance, but if you want to estimate heritability, or you want to understand correlations between different types of relatives, then you need to have a model for the correlations between distinct pairs of individuals. And while the sort of random effects approach, it treats genotypes as observed, and it kind of treats the observed genotypes as the causal genotypes in some sense. And the effects is random, which is something like a Bayesian prior or whatever. But the more traditional quantitative genetics way of analyzing correlations between relatives is to treat effects as fixed, which means you don't have to make assumptions about the distribution, and to treat genotypes as random. So you're considering like the correlation or covariance between some unobserved causal genotype for a pair of individuals. And it's a question, what does that covariance in genotype depend on for a pair of individuals? And it depends on the identity by descent sharing between those individuals. So identity by descent is really saying, like, do you have segments of DNA, homologous segments of DNA that are inherited from the same recent common ancestor? So I'm just illustrating it here for siblings. So if you consider, this is just say like the paternal side or the maternal side, so this would be the father or the mother. They have 2 homologous chromosomes here in 2 different colors. Then you have meiosis producing the inherited chromosome for one sib, and a different meiosis producing the inherited chromosome for another sib as a mosaic of the parental chromosomes. And then here they've both inherited like the pink one, so that's IBD because it's identical by descent from the parents. And in the blue bit, they both inherited the blue one here, so that's also IBD. And you expect 1/2 of a chromosome for siblings to be IBD. And it's more complicated for other relatives. And this is sort of the basis of deriving the correlation between sort of unobserved causal genotypes, because you imagine them to be randomly distributed in the genome with respect to identity by descent sharing. So what you do is you assume that the genotypes are identical when they fall in an IBD segment, which isn't exactly true when you have mutations, but they're pretty rare, so we don't worry about that too much. And then you assume that they're independent when they don't fall in an IBD segment, which is also a somewhat questionable assumption. But it works pretty well. And this gives you the covariance between a pair of individuals' genotypes as the variance of the genotype, where F is the allele frequency, multiplied by the relatedness coefficient between the pair. And this relatedness coefficient is twice the kinship coefficient. And the kinship coefficient is basically saying if I randomly select an allele from one individual and I randomly select an allele from another individual, what's the probability that they fall in an IBD segment? So to do that, you have to average over the 4 possible haplotype pairs from one from one individual, one from another individual. So it's the average over the paternal-paternal IBD sharing, the paternal-maternal IBD sharing, the maternal-paternal IBD sharing, and the maternal-maternal IBD sharing. And you divide that by 4 to give you the kinship coefficient, which is this probability, and then you need to multiply it by 2 to scale for the fact that things are diploid and additive in this model. And that gives you the covariance between individuals' diploid genotypes. So I'm not going to go into all the details, but this R is like so you have a phenotypic covariance matrix here, and then this R is like a matrix of the relatedness coefficients between individuals derived from identity by descent sharing. And this decomposition of a covariance matrix is related to the variance decomposition I derived before from considering the splitting of environmental component into a parental component correlated with parental genotype and uncorrelated. So you can see how these different components of phenotypic variation contribute to the covariance between pairs of individuals in a population. And the covariance between a pair of individuals due to the direct genetic component is given by the relatedness matrix, the relatedness coefficient that I derived before. So that's the sort of like relatedness between the offspring. And then the covariance between a pair of individuals due to this sort of gene-environment correlation bit, the bit that's correlated with parental genotype, that's given by a generalization of the relatedness coefficient to relatedness between the parents of one individual and the parents of another individual. And it's actually the sum of the kinship coefficients between the 4 parent pairs between two sets of parents. Then the covariance between individuals due to the covariance between genetic and environmental effects is given by this relatedness coefficient, which is like the average of the relatedness between the offspring of one family and the parents of another, and the offspring of the other family and the parents of the other. So this doesn't completely determine all the possible sources of covariance between a pair of individuals and a population, because there can still be covariances of this residual environmental component that can be due to sort of random family environment effects or population stratification. So there's still an unknown environmental covariance component in this decomposition of the phenotypic covariance matrix. So that can be problematic potentially if you don't think about it.
Alex?
Yeah.
Right here. So the first term in your decomposition is the one that that I think you're focused on, because it's like the direct effect heritability and stuff. Are there intuitive ways that we might be interested in your second and third terms beyond, like, I mean, it sounds like you're treating them a bit just like nuisance parameters that we need to include, but do you think there's interesting ways to interpret those as well?
Well, I mean, yeah, I mean, this one is pretty interesting, I think, because it gives you the covariance between genetic and environmental effects. So are genetic and environmental effects highly correlated? That's what that term tells you. And this one's sort of telling you, like, what fraction of the environment is actually related to genetics. So they are interesting components, but estimation of those components is currently rather tricky. But, well, I'll go into that a bit more later, but I did get estimates of all these components. I didn't really emphasize them in my paper because they do pick up, I don't know if I want to go into all of that, but because there's unknown residual environmental covariance, these can pick up bias from that, whereas the direct component doesn't. So the estimation of these components is trickier, but so you can sort of see them as a nuisance parameter if you're just interested in heritability, but they are of interest, but actually estimating them accurately is challenging. Yeah.
Alex, I was wondering, so we had a discussion yesterday about this relationship between effect sizes and heterozygosity and how that basically determine the genetic architecture of certain traits.
Mm-hmm.
So just wondering, how can you pick that up in that framework?
Well, I mean, in the way that I've derived it here and in the main analysis in my paper was based on IBD sharing, which is sort of agnostic of the relationship between, it doesn't make assumptions about effect size and relationship between effect size and allele frequency. So that wouldn't, this method based on IBD sharing wouldn't worry or infer anything about that. But I mean, if you do this, you can do a SNP version of this, which then has all the whatever issues, Gremel and LD score, where you have to make assumptions about these things.
Are you saying that if, regardless of those assumptions, that this approach will always give an unbiased estimate, even if the sort of model is different?
Well, it doesn't make any assumptions about the distribution of effect sizes. So I mean, it's a bit like the twin thing, right? The twin thing's based on IBD sharing, and or the SIB regression thing, and like, yeah, no one really worries about effect size distributions when you're doing twin estimates, right? Because you're not making assumptions about effect size distributions. That's one of the advantages of IBD-based methods, is you treat genotypes as random and effects as fixed, and you don't make assumptions about the effects. So that's the sort of background theory, and then you can think about how would you actually estimate these parameters, especially the heritability, which is the easiest one to estimate, but as I was saying, the other ones could be interesting to estimate too. So I'm just going to briefly talk about different methods of estimating heritability. So heritability analysis traces back to Sir Francis Galton, cousin of Charles Darwin, and I found recently that not only does my academic work trace back to Galton, actually my academic lineage traces back to Galton through Peter Donnelly and Pearson and whoever, so it's kind of an amusing fact. But so Galton wrote this book, Hereditary Genius. He wrote a lot of other groundbreaking things in statistics and genetic analysis. But this book, Heterogeneous, set out one of his main sort of contributions to thinking about heritability and the role of genetics in behavior and achievement and things like that. So in this book, he developed a method that said, do individuals that achieve eminence tend to have relatives who also achieve eminence in the same field? And he looked at like various fields like art and science and literature and, like, bizarrely enough, wrestling, which I'm not sure why I said that. But the basic idea, in some sense, is similar to a lot of methods for estimating heritability. Is you're looking for a relationship between phenotypic similarity and genetic similarity or relatedness. You didn't have any genetic data; it was just based on pedigree relations. And you're saying, are people that are related more likely to both be eminent in a field? And he found some very strong enrichment for eminence among relatives. However, this could obviously be driven by environmental sharing and not necessarily by genetic sharing, so it wasn't a very well-controlled method, but it certainly was very influential in making people think that these hereditary factors, they didn't even know what genes were. They didn't have a Mendelian model of inheritance then, but it was certainly an argument that hereditary factors contribute towards human achievement. So that's sort of the key problem or difficulty in estimating heritability, is that these methods tend to look at how phenotypic correlation changes with relatedness. But as relatedness increases, so does environmental similarity. So simply looking at how phenotypic similarity changes with relatedness in general is going to be biased by environment if environmental similarity increases with relatedness. And the different methods of estimating most different methods of estimating heritability can be thought of at a high level as fitting some sort of linear relationship to subsets of points in this space. So the twin estimate looks at the difference in correlation between MZ and DZ twins, and that is the gradient of this blue line here. But if MZ twins have more similar environments than DZ twins, then they'll be overestimating. So more recently, these sort of Gremel methods, which I'll say a bit more about later, become popular. And they look at how, at a high level, how phenotypic similarity changes with relatedness estimated from SNPs for distantly related pairs of individuals. But if environmental similarity increases with SNP relatedness for distant pairs, then they can be biased. And when you have indirect effects from relatives, and sort of you're saying if your SNPs become more similar, then your parent SNPs become more similar. So that's going to track the indirect effects from parents in the same way as it tracks the direct effects, essentially. I don't know what, my slide's not working properly here. But then a method that is perhaps less well known or less commonly implemented is one I call the kinship method. And this looks at how phenotypic correlation changes with relatedness estimated from IBD segments for all pairs in population, close relatives and distant relatives. The problem with that really is it has no principled way of distinguishing environmental from genetic effects, and you can have a lot of increase in environmental similarities. You go up the relatedness spectrum, and especially indirect genetic effects that really can't separate because they increase in proportion to relatedness, the correlation of indirect genetic effects. I think I'm just going to use this. Okay, then there's this method developed by Peter Fisher in 2006, and this takes advantage of this random segregation within families that I've been talking about. So siblings vary in their relatedness. As shown before, you have these meioses, and they can share IBD, they can not share IBD, and how much IBD they share or not, and there's deviation around the expectation, which is like 0.5 for the relatedness between siblings, this variation around the expectation that is there for siblings. That's uncorrelated with almost all kinds of environmental effects. So if you look at how phenotypic similarity changes with relatedness for sibling pairs alone, you can estimate heritability in a way that is robust to environmental effects. Apart from indirect genetic effects between siblings can be an issue for this method, but apart from that, it's pretty robust. The main issue is it needs like hundreds of thousands of genotype sibling pairs to get precise estimates. But you can do something somewhat similar to the sibling regression method. This is the method that I developed when I was in Iceland. So if you think about this phenotypic covariance decomposition, you still have this unknown covariance of the residual environmental effects, which can generate bias potentially. But actually, it turns out if you just pretend that this is equal to the identity, in other words, there's no covariance of the residual environment between distinct pairs, then you can, fitting this covariance model, you need these 3 relatedness matrices derived from IBD sharing between parents and offspring. You can estimate heritability in a way that is robust to environmental effects. You can sort of prove that theoretically. And that the sort of key idea underlying that is this phenomenon of relatedness disequilibrium, which is a consequence of this random segregation of genetic material in the family. And the basic idea here is this relatedness between the parents. So you have the relatedness between one set of parents and another set of parents. Then based on the laws of Mendelian inheritance, you can compute the expected relatedness between the offspring of one set of parents and the offspring of another set of parents. And that's what I'm showing here. It's the relatedness between the parents. This diagonal line gives the expected relatedness. And then this is the actual relatedness. So any deviations around this diagonal expectation line are generated by the random segregations in the parents. And those random segregations in the parents are independent of almost all environmental effects. And that means that if you use, instead of just using the relationship between phenotypic similarity and relatedness, you instead use the relationship between phenotypic similarity and the deviations around the expected relatedness given the parents. One can then estimate heritability in a way that's robust to environmental effects. And the SIB regression is really a generalization of the SIB regression thing, because this is like the siblings, and then all this variation is like vertical. I mean, there's sort of inbreeding and things, we don't worry about that. But the siblings, the information of the SIB regression is this variation, and then this method is sort of generalizing it to all pairs of individuals, which gains you a lot more power, because actually most pairs of individuals in a population are distantly related. So that was the method that I developed, and it was published last year. I'm just going to sort of show some of the main results. So the method is relatedness disequilibrium regression. It comes from fitting that covariance model. And by fitting all those relatedness matrices jointly, you're essentially using the random variation relatedness within a family rather than just the variation relatedness, which can be confounded with all sorts of environmental things. Then you can see, comparing it to like the methods that just look at the relationship between phenotypic similarity and relatedness, you see that they're overestimating things like height, educational attainment, body mass index, age of first child in women. So that's evidence that there is this confounding between genes and environment, between relatedness and environmental similarity. If you compare to twin studies, then you see a big difference across all the traits. Basically, the caveat here is that the twins are from Sweden and my analysis is from Iceland, so it'd be nice to do the method in the same population, but the Icelandic population is so small there aren't even enough monozygotic twins to do a proper twin study. Yeah.
So if we think that there were non-additive genetic effects, then the twin studies are estimating some mixture between the narrow and the broad sense heritabilities. Is RDR estimating the same mixture?
No, so RDR basically just estimates the additive components. I did simulations with like epistasis and dominance, and it picks up a tiny fraction of it, but not much.
So this may not be about confounding. Like this might just be because you're getting narrow sense rather than.
So I think that the non-additivity will contribute to some of this difference, but unless non-additivity is like quite high, I don't think it could explain it all. And I don't think there's a lot of evidence that it's high enough to explain the gap. I don't really know what the explanation for the gap is, but the gap isn't just present comparing like the RDR methods to twin studies. Like if you can say, oh well, my method's this new complicated method, like it could be going wrong in all sorts of ways we don't understand. But the SIB regression thing is like very simple. Everyone thinks they understand that at least. And if you compare that to the twin estimates, you see a very similar pattern, apart from perhaps educational attainment, but I think the SIB regression thing is biased by the indirect effects between siblings, so that might explain that. So this is a question I think needs answering, like there is this gap between twin and genomic-based methods, and I don't think anyone really knows the explanation for that yet.
So Alex, you're using, you use unrelated individuals?
Sorry?
You use unrelated individuals for RDR? Like
No, you use everyone.
You use okay, so you don't do like just Cornell where
You could do that if you wanted, but there's no need to.
I see.
You would lose a lot of your power.
Yeah, well, okay. And have you could also use RDR or an equivalent with extended pedigree data, right? Like just like, I mean, basically fitting this with half sibs and sibs and all that, right?
That doesn't have the same theoretical guarantees. I thought about that, but the covariance term is not properly controlled for with extended generalizing it to like pedigree. It's going to control for it to some degree, but it doesn't control for it properly, perfectly. No, it's kind of a complicated explanation. We can talk about it later. So then I talk about grammar. So I mentioned something about that before, that it's like at a high level is looking at how phenotypic similarity changes with this sort of SNP similarity or SNP relatedness for distantly related individuals. And this is a high level sort of understanding of it, but that high level understanding of it comes from like the consequences of the modeling assumptions of grammar. So you model an individual's phenotype as the sum of these genetic effects, like additive genetic effects. And you use normalized genotypes, which has some important consequences we were discussing a bit yesterday. And then you assume some sort of environmental sort of noise on top of that. So it's basically like regression on a big set of genetic variants genome-wide. And to do that, you have to make some assumptions. So the standard assumption is that the genetic effects are normally distributed. So as we were discussing yesterday, I like to think of this as like a prior on the genetic effects, but because of this assumption and the way the genotypes are normalized, it does imply a particular relationship between allele frequency and effect size, and that the effect size goes up as the allele frequency goes down in such a way that the expected variance contribution from any SNP is the same irrespective of the allele frequency. And that assumption has been shown to generate some biases, and there are different ways of accounting for that by, as Patrick was saying yesterday, partitioning by NAF and LD and all that sort of thing. But I still think the IBD thing has the advantage of not having to deal with all that. But the IBD thing does, I'll show that in a sec actually. But then yeah, the environmental effects are assumed to be like uncorrelated between distinct pairs of individuals, and you know that might be true, but it might not be true, especially if you have population stratification that has not been properly accounted for, adjusted for by principal components, and as we've been discovering recently, principal components are not really as effective at dealing with all sorts of stratification as we would like. And then these modeling assumptions lead to a Grammel model for the phenotype. So I'm just assuming the mean is 0, but you can model mean effects. That's not really a problem. A Grammel model of the phenotype distribution where you're modeling the phenotypic covariance between distinct pairs of individuals is proportional to this SNP relatedness between the pair, where the constant of proportionality is giving you the variation in some sense explained in a regression sense by all of the SNPs that you put into the Grammel model. And the SNP relatedness can simply be thought of as like the average correlation between a pair of individual SNPs across the genotyping array or whatever information you put into it. And then the Grammel SNP heritability estimate is just the ratio between the estimate of this genetic variance parameter and then the sum of the genetic variance and the residual environmental variance parameter. So that's trying to give you it's like the fraction of variance explained by this regression, what you could hope to explain by a regression on all of the SNPs genome-wide if you had like an infinite sample size. That's the way that I think of it, at least, rather than like heritability. It's different to heritability in my opinion, but people call it heritability, and it is perhaps similar to heritability under certain conditions. So I talked about IBD. So the IBD relatedness coefficient is different from the SNP relatedness coefficient. Although actually, if the number of independent SNPs went to infinity, they would actually converge to the same thing. So this is the relationship, and you actually see for closely related individuals where the IBD relatedness is high, the IBD relatedness and SNP relatedness are highly correlated. But if you go down to people that don't show any IBD segments, which is like most people in a population don't show any detectable IBD segments, then there's still a lot of variation in SNP relatedness. And this is actually why the sort of Grammel SNP-based approach has a lot more power than the IBD-based approach. It's sort of a bias versus variance trade-off to some degree. It has a lot more power than the IBD-based approach because within segments of DNA that are not IBD, there's still variation in how similar people's SNPs are. So the Grammel SNP approach is taking advantage of that to estimate the variance explained by those SNPs, but it can only estimate the variance explained by the causal SNPs that are in linkage disequilibrium with the SNPs put into the Grammel model, whereas the IBD-based approach can theoretically recover the full genetic component of a trait because it's sort of covering the whole genome in the unobserved causal variance and whether they lie in IBD segments or not. But it has lower power. So then there's a question of what is like Grammel actually estimating, because it's not necessarily the heritability, because it's basically like a high dimensional signal to noise ratio, like the amount of variation you could hope to explain with like a regression infinite sample size or something. So I did some simulations, unrelated individuals, heritability, SNP heritability 20%, and then a parental genetic nurturing effect 1/3 of the size of the direct effect. And then because you have this sort of magnification thing with the square, with the effect size, this means that the total variance, so they're perfectly correlated to the direct and indirect effects here. This means the total variance explained by the combined direct and indirect effects of the transmitted alleles is 35.56%, so a lot more than 20%. So then the Grammel estimate is basically estimating that. It's the total variance explained by the direct and indirect effects of the transmitted SNPs, which makes sense because that's sort of what you're getting when you regress on a SNP. And that I've derived theoretically as well and related it to that variance decomposition. So you're getting the direct component, you're getting 1/2 of the component that's correlated with parental genotypes. You only get a half because only half of the alleles are transmitted to the offspring. And you also get the covariance between genetic and environmental effects. And if you fit PCs and whatever, you can remove some of the stratification, but you basically can't remove the indirect genetic effects from the parents, from the Grammel estimates. If you do the SNP version of my method, then you can separate out those components. So, I mean, yeah, I should say that like this component that Grammel's estimating, it's not the heritability, but it doesn't mean that it's not interesting or useful, because it is something more like an upper bound on prediction that you could hope to achieve. But in terms of heritability, it's not the same thing as what you might get from twin studies. So comparing like Grammel estimates and twin estimates is a bit fishy, in my opinion, especially for traits like educational attainment. So if I compare the SNP version of my method to sort of Grammel, this is like a Grammel type method. It's a little bit of a different inference procedure, but it should have the same sort of expected value as the Grammel approach, or with a very large sample size at least. Then you can see that this Grammel type method is giving higher estimates than the SNP type method for some traits, including educational attainment. It's like 70% higher. Age of first child in women, also about 70% higher. And height, it's a bit higher as well. And there's probably some contribution of assortative mating to this, but at least for educational attainment, this ratio of the two effect estimates is very consistent with what we estimated is the indirect genetic effect for from the polygenic score, and it's also consistent with some of the within-family analyses done in EA3, although actually they seem to be estimating a much stronger indirect genetic effect than we had in Iceland. So there's a question of whether that's because of the British class system or something, and Iceland's a very egalitarian country, so maybe this is smaller. And then this brings me on to like, it's something I've been thinking about recently, which is that, you know, people often say, oh, heritability is an upper bound on genetic prediction, and it's not. When you have gene-environment correlation, that's not an upper bound on genetic prediction. You have to specify what you want to predict, right? I mean, in some sense, if all your goal is to predict with maximum R-squared within the same population that you got your sample from, you can leverage population stratification. Even if you don't want population stratification to count in your prediction, you can definitely leverage indirect genetic effects from relatives. So the actual maximum possible prediction accuracy that's kind of what Grammel is potentially estimating, and that can be larger than the heritability. So I was actually thinking, like, what would it be for educational attainment? So if you take this 1.7 factor, which is like the inflation relative to like the heritability, and then you take my estimate, the RDR estimate of heritability is like the lower bound, and then the twin estimate is the upper bound, and you actually see that like maybe, I mean, this is just a rough sketch of what might be possible. You see that the maximum possible prediction accuracy for educational attainment could be up to, if the twin estimate is right and the strength of indirect genetic effects is estimated as right by us, then it could be that the maximum prediction accuracy for educational attainment is up to 70%. Seems a bit high to me, I have to say. It's like there's no justice in the world if that's true. But 34% seems a bit low to me, so my guess is it's somewhere in that range though. Then there's the theoretical context for all this. So one of the interesting... Yeah.
Go back one slide, please. Does the 68%, is that by inflating the twin estimates by 1.7?
Yeah.
But the twin estimates shouldn't be biased by these indirect effects, should they? Because the twins come from the same...
Well, exactly. So if the twin thing is really getting just a direct component, then it's missing all of the component that's coming from the indirect effects and the covariance between them. So that's why you inflate it. So theoretically, the twin thing is underestimating the potential of genetic prediction when you have indirect genetic effects, which I think is an underappreciated point. So everyone thinks the twin thing is like the maximum possible prediction. But when you have the indirect effects from relatives, it can be much higher than the twin estimate potentially.
Just to
Yeah.
to be realistic on that previous slide, like we're not seeing, with our actual prediction, we're getting, it's looking like it's going to asymptote at like 15% in the meta-analysis, and if you believe the Gremel estimates, it could get up to like 20% in. So it seems like it's going to be more like the lower bound than the upper bound.
Well, that would probably imply that the twin thing is overestimating then, I think.
Yeah.
Yeah, well, that's only on like common SNPs, I guess, right? So theoretically, you could go.
Twin estimates are for all sequenced and all the interactions, like so if we had infinite time and infinite data.
Yeah, so this is not a realistic level of prediction, but it's sort of a theoretical maximum that if you had perfect estimates of all effects of all genetic variants.
Got it.
Yeah, this is also of all variants, including rare variants?
Yeah. I mean, yeah, this isn't like a paper or whatever. It's just like a thought experiment to make you realize that it can be more than the heritability. So one of the interesting things when you have these indirect effects from relatives is like the trait distribution is not stable over time. So if you imagine a population that didn't have indirect effects and you injected them in, the trait distribution would have to reach some sort of equilibrium because you're accumulating. You have the indirect effect from the parents onto the offspring, but then there's an indirect effect on the offspring, and then they have an indirect effect on their offspring. So you have one way to think of it is that your phenotype depends on the genes you inherit, but also the phenotype of all of your ancestors in a geometric decaying sequence. And the theory of all this is quite cool, and like I had fun deriving like a bunch of stuff with it. And then I discovered it had all been done in the 1970s. So there you go, by Cavalli Sforza, one of the founding fathers of modern population genetics, and Feldman, who I'd actually meet and talk to about this stuff. And I think this theoretical work that was done in the 70s and then taken on by other people, and it's used in the field of social evolution quite a lot, is something that I think people interested in the social sciences should take seriously, because it's quite a sophisticated theory of like combined cultural and genetic inheritance. And some of the theory includes like assortative mating and selection and all these different population genetic things. So this is sort of a richer theoretical framework for understanding traits that are partially culturally mediated. And it gives you some sense of the like population genetic cultural processes that are generating like social and health inequalities, and the sort of standard quantitative genetic models, which are more derived from animal breeding, where there isn't this kind of like accumulated cultural inheritance pattern. That's all I wanted to say.
So I just have a question about what are the requirements for doing RDR in terms of sample size, but also what variables you need?
Yeah, well, you need genotype. Well, the version that I've published is you need genotype information on both parents of each phenotyped individual, and you would need a fairly large sample like that to do the IBD version and have any power. So the smallest sample that I used was for educational attainment. Unfortunately, that was the smallest sample, and that was like 10,000 people with both parents' genotypes. So I think for the IBD version, and that's also in Iceland, so there's more variation and more relatedness. So I think you probably want a minimum of 10,000 for the IBD version. I think you can get away with smaller sample size for the SNP version, because there's a lot more power in the SNP version. So maybe, I don't know, 5,000, at least a few thousand though, with both parents typed. I did actually develop a version that only requires one parent typed. I derived loads of estimates from that, but then I got kicked out of Iceland, so I don't know if it's okay. But so you can do it with one parent type, but it's like it's kind of a lot more complicated.
Okay, I was going to ask you.
Have you compared, like in real data, how the SNP relatedness and that IPD relatedness leads to difference in the heritability estimates? I think you had a page from simulation, but I was wondering if you replace the R in your method with the Gremlin GRM. What is the heritability estimate?
So you mean the difference between the SNP version and the IVD version of RDR? Or the difference between like the Gremlin and just like using IVD instead of the Gremlin thing?
I think I mean the latter actually. You replace the R in the RDR method with Gremlin GRM.
So yeah, the SNP version of RDR, and it is kind of that is a complicated comparison because the RDR thing's using close relatives, and in that case the SNP relatedness and the IVD relatedness are not that different. So that comparison is not like completely clean, but you can do that on the basis of the numbers in the paper, and you do see that the IVD one is bigger for almost all the traits and quite a bit bigger for some traits. It's actually a lot harder to make that comparison between SNP and IVD estimates, and I think people realize because the IVD thing's like picking up a lot more structure and stratification and different. They have differential confounding with like environment and things like that, and then differential picking up of rare variance. So you can make the comparison, but it's hard to interpret it in a clean way, I think.
So I have a question about your idea about how genetic nurture from siblings may lead to the violation of the core environmental assumption in twin estimate, because I thought that from our earlier discussion, those variations can be part of the genetics, so it is not really a violation of the.
Well, I think it depends on what you want to define the heritability to be, but if the twin thing is picking up to some degree the indirect effect from the sibling, then it's picking up part of the family environmental influence. So that wouldn't traditionally be part of the heritability, but it is genetic in some sense. So, but actually the relationship between the indirect effects from siblings and the bias in the twin estimator is actually quite complicated. So it's not that the twin thing is then estimating an easily interpretable parameter in terms of the direct effects, heritability, and the indirect effects. I mean, I think it is interesting to also think about other variance components like the Gremlin thing, which is picking up the indirect effects to some degree as well. But then you could also define something like the total variance explained jointly by the parental and the offspring genotype, which would be like a family trio heritability or something. So, you know, you can define different variance components that are interesting for different questions. Doesn't mean they're easy to estimate, though.
So you said you need to have parental genotype. I wonder if you ever had data with parental phenotypes as well, and if that helped in any way with educational attainment.
Yeah, so we did do an analysis in the Nature of Nurture paper where we fitted parental education jointly with the parental polygenic score, and you see that the parental polygenic score effect, like the indirect effect estimate or whatever, shrinks by about a half. So that's evidence that part of the indirect effect from the parents captured by the education polygenic score is mediated through a parental phenotype that's correlated with parental educational attainment, but it doesn't seem to be capturing all of it. And there was a recent paper from some people at King's where they did something similar, where they looked at polygenic scores, and then it's a bit more it's a bit different what they did. They did with DZ twins, but they did see that the sort of indirect effect component of their analysis shrank a lot when they fitted parental socioeconomic status for cognitive and educational traits. So clearly part of the indirect effect is being mediated through those family level variables of education and socioeconomic status, but it's unlikely to be all of it. Like the parental phenotype, actually another point is that using the polygenic score method actually probably underestimates the contribution of the total contribution of indirect genetic effects, because The polygenic score has been ascertained based on GWAS effect estimates that combine direct and indirect effects, so it's sort of biased towards the direct effects. And you can also have indirect effects from parents that go through phenotypes that are genetically uncorrelated with the offspring phenotype. So the polygenic score approach is not capturing the full indirect component from the parents. The RDR variance component would be theoretically, but they can also be biased. And like, yeah, I think it's an interesting question to see like what parental phenotypes are like mediating this kind of effect, and I don't think we fully know, and it's impossible to measure. It's like all of the nurture you get from your parents from the moment you're conceived, you know, in some sense. So it's like an infinite dimensional phenotype.
But what are the estimate how much of that effect could be? Because there are a lot of, or not a lot, but there is some research in social sciences where you just observe phenotypes and you kind of regress parental education, or you control for parental education when you estimate children education. So how much of that effect, according to your studies, is probably mediated by the genetics?
Well, it depends. I mean, it depends what you mean by mediated by genetics. I mean, the sort of correlation between parents and offspring for a trait is confounded with the genetic transmission to the child. But then the parental phenotype itself can be partially genetic. So you could try and measure like the heritability of whatever parenting behavior, right? To see that would tell you something related to this. I mean, I think it's interesting that you can potentially use the genetic information in conjunction with the parental phenotypes to try and get a better sense of what is and isn't confounded in those kind of studies, of like parenting but effects on education.
Yeah, I was actually wondering, is there, so since you have like a fairly large data set, did you try and like look at them sort of binned by age group essentially, because almost like a longitudinal kind of stratification, maybe, because I was just wondering if you could actually separate out the effects of, you know, you have a parental environment, but technically at a certain point, you imagine children are emancipated, and they go out, and then there is a change in the environmental condition associated with that. So I know that some traits it doesn't necessarily carry over, but if there is a way of actually using the spread across ages.
Yeah, well, I know twin studies have done a lot of stuff like that, where they measure heritability at different ages, and they do see that it changes. Like I think the shared environmental component estimated by twin studies for like educational attainment is a lot higher when children are young, and it decreases. And I'm sure you could do something analogous with these methods, but I don't think the power is there yet. I mean, even with the big sample I had in Decode, like the estimates are fairly precise as far as heritability estimates go for some traits, but for educational attainment, for example, like using all the data I had, I still only had like 10,000, so you don't really have any power to start like decomposing that into different demographic groups.
Okay, so we'll resume